Okay, hello everybody. Good evening. Welcome to Macquarie University. My name is Linda Beaumont and I'm Deputy Dean of the Faculty of Science and Engineering. This is our November Discovery Lecture Series. It was to be the last one that we would be running this year, but we will actually be having one more in December, which our outreach team will tell you about at the end of this. So to begin, I'd just like to acknowledge the traditional custodians of the land that we're on, the Wallumattagal people of the Darug Nation, and pay respects to elders past and present. I'd also like to extend that respect to any Indigenous people who are with us today. So throughout this year's Discover Lecture Series, we've explored the remarkable ways in which science and technology shape our world and our future. We began with the rise of artificial intelligence, its power, its pitfalls, and the potential to transform humanity. Then we dived into the unseen universe of the gut microbiome and how the microbes within us influence our health and disease. We heard how cutting-edge research at Macquarie is revolutionising scam prevention through AI-driven innovation. And we journeyed from the sandy origins of microchips to the global supply chains that power modern life. Our speakers have challenged us to imagine greener, more resilient cities. They've guided us through the evolutionary histories of primates, and they've revealed how microbial genetics can tackle global problems. Together, these talks have celebrated the curiosity, the discovery, and the transformative impact of the research that we're doing at Macquarie University. And tonight we have our second last speaker in this series. It's my honour to introduce you to Professor Rick Shine. Now I just want to give a bit of a blurb as to Rick's career. His career spans nearly 5 decades of influential research in evolutionary biology and herpetology. He obtained his PhD from the University of New England, where he studied the ecology of Australian venomous snakes. After some postdoc positions in the US, he returned to Australia to work at University of Sydney, where he became a professor in 2003. Following that, he enjoyed a series of ARC fellowships, becoming a laureate at some point in that journey. His early research focused on the ecology and evolution of reptiles, especially venomous snakes. He conducted global fieldwork on snakes around the world and, if Wikipedia is correct, and you can let me know here, Rick, dissected thousands of museum specimens to document the natural history across continents. Rick established long-term ecological studies in Australia on lizards, endangered snakes, and a host of other species. The expansion of the geographic range of invasive cane toads transformed his research, leading to major discoveries about toad impacts, innovative control methods, and the forces behind their accelerated invasion front. His team pioneered with taxon-baited tadpole traps, suppressant pheromones, and more recently, gene knockout Peter Pan tadpoles using CRISPR-based approaches. After moving to Macquarie in 2018, Rick has continued to advance conservation-focused research, including novel tools for protecting native species. I'd like to summarise Rick's career with a couple of numbers and a couple of names. 1,100-ish, 3, 2003, 2005, 2015, 16 and 19, Clark, Wilson, Fitch, Mueller, McFarlane, Burnett, Walter Burfitt, Robert Whittaker, Prime Minister, Dmanzia Shinai. What is all of that? Does that make sense to anybody? 1,100-ish is the number of publications that Rick has co-authored. Rick, at any given moment, do you actually know the number, or is it changing so rapidly there's no point now trying to recollect?
I'm not very good at counting.
3. We have a number of scientists in here. Would anybody be happy with just one eureka? Yes? Rick has 3. 2003, he was elected the Fellow of the Australian Academy of Science. 2005, a member of the Order of Australia. 2015, he was elected President of the world's largest herpetology society and a Fellow of the Royal Zoological Society of New South Wales. 2016, New South Wales Scientist of the Year, and received the Prime Minister's Prize for Science. 2019, and I'm leaving lots of gaps here. They aren't really gaps though. There's many other things in between all of those years. 2019, elected Fellow of the American Academy of Arts and Science. Clark, Wilson, Fitch, Mueller, all of those names that I read out are just some of the incredible, illustrious awards that Rick has received over his career. Demansia Shine. How else, I mean, what would top a career on venomous snakes and invasive species other than having a venomous snake named after you? Rick is lucky enough to have achieved that. So in what I'm sure is a very interesting talk, can you please put your hands together and welcome Rick Shine?
Thanks very much, Linda. I'm hoping that the microphone's working, and please yell at me if it isn't. So Linda has set up your expectations far too high, but I want to start by thanking you all for coming here and missing out on that gorgeous late afternoon sunshine that's just out there in the world out around us. But in particular, I want to thank three people that are here who have played a really major role in the toad research and continue to do so. We've got Michael Clark and Alex Paparakis. Wave your hands in the air or something, guys. These are the sort of the genetics gurus that are doing all the stuff that I don't understand about CRISPR that you're going to hear about towards the end of the talk. And my wife Terry has been the unpaid volunteer for decades. 2 weeks ago we were at Daly Waters chasing cane toads. It was 45 degrees every day. So Terry really does deserve sainthood. So the creature that doesn't deserve sainthood, according to most Australians, would be the cane toad. And you really don't have to worry too much about its scientific name because these things do tend to change. But you need to know that this is a really big frog, and you know, you can have a look at these guys later. These are reasonably sized ones, but they can get huge, like the one in the picture. You'll also note that it has very large swollen area on its shoulders, and these are the poison glands that produce a really distinctive set of chemicals like no other in the world, pretty much, certainly like no other frogs. And these are incredibly powerful toxins. So, you know, this girl here, the toad, the lady is probably non-toxic, but the toad would have enough toxin in those glands to kill just about everybody in this room, I would think, very quickly, within a few minutes of a heart attack. It is incredibly powerful toxin. So, the other thing you need to know about the toad is that it really truly is one of the most well-travelled frogs on the planet. There was actually a rush of articles on some of the internet sites last week calling it the world's most invasive species, which is nonsense, but still pretty cute. The story is pretty straightforward, that we have this giant frog living in the Brazilian rainforest in a large area in northeastern South America. And in the 1800s, people that are growing sugarcane in the Caribbean are having a problem with beetles whose larvae are eating the roots of the sugarcane plants. And somebody sees these toads hopping around and thinks, look, they're big and they're hungry and they eat a lot. And so we'll bring them to the sugarcane plantations and we'll let them go and they'll control the beetles for us and that will increase our yield of sugar. And so they were moved around the Caribbean a bit, ending up in Puerto Rico as well as other places. And in general, you know, people thought they were probably doing a pretty good job, but nobody ever really gathered information on it. And in 1932, there was a sugarcane growers conference in Puerto Rico, and an American named Cyril Pemberton was convinced of the joys of the toad. So he brought 150 cane toads all the way from Puerto Rico to Hawaii, where they were released in the sugarcane plantations. They liked Hawaii, as most people do, and they began to breed. And 3 years after that, the Queensland government sent a young Australian bloke named Reg Montgomery off to Hawaii so that we could have our own cane toads. Reg brought them back to Australia, 101 adults. His mate said, build a nice little pleasure palace. I don't think I have anything to point a, anyway, what the hell. On the top right, it's a nice little pleasure palace. The toads bred there. And so before very long, there were thousands upon thousands of baby toads, and they were released up and down the Queensland coast to great acclaim. People thought that this was a fantastic, environmentally responsible form of pest control. Didn't require poison or anything like that. The toads were going to take care of the problem. Everybody was quite confident that a giant frog from the rainforest would never move anywhere else in Australia, because the only rainforesty areas are along the east coast of Queensland. But of course the toads had other ideas. And this is the most recent map of this year. So the toads now cover a large part of tropical Australia. They'll be at Broome on the west coast within a year or two. So they've gone all the way across the continent, which is not bad for a short, fat little amphibian with stumpy legs. They've mostly done it on their own. They do get lifts from people. They stow away in caravans and landscaping materials on trucks and so on, but the toads have pretty much done it themselves. So we now have a very large amphibian in Australia, a true toad, and of course there were no true toads in Australia before this happened. Australia was so separate from the rest of the world that toads never got here. There are toads in Asia, there are toads in Europe, there are toads in South America, there are toads in North America, there are toads in Africa, but there were no toads in Australia until 1935. So, as Linda said, I was a snake biologist, and I was having a great time studying snakes, and particularly I had a long-term study area near Darwin, between Darwin and Kakadu, near a town called Humpty Doo that some of you have doubtless driven through. And the toads weren't supposed to get to the Darwin area for another 40 or 50 years, but they accelerated for reasons that we ended up doing a lot of research on and ended up being interesting. But anyway, 2005, you could sort of, you could look to the east and you realised that the toad invasion was coming and it was going to be upon us quite soon. And according to a lot of the stories that people were telling, it might wipe out the snakes that I'd been studying for a couple of decades up there on the floodplain. And it was clearly an incredible opportunity. We had this really intensely researched chunk of tropical Australia. That research had been paid for by the Australian taxpayer. And here we had a high-profile pest that was about to arrive and maybe demolish part of that ecosystem. It was clearly an opportunity to find out what the hell an invasive species is doing and how you might go about doing something about that. And so I expanded the team. We kept playing with snakes, but we put more and more of our effort into toads. That's an incredibly loud air conditioner. What the hell? Okay, very, very brief summary. People worry a lot about the ecological impacts of cane toads. Before we started work, there was a strong feeling that the toads would eat all the bugs and that would affect the ecosystem. They would compete with frogs. They would spread parasites to frogs and so forth. None of that turns out to be really all that important. This male green tree frog is having a good time with this female toad at our research station, so he might have had a bit more variety in his love life because of the arrival of cane toads, but broadly, the toads have not really affected the prey base or the native frogs. What they have done is they've slaughtered the predators. The problem is that we have no native toads in Australia and that the poisons of toads are really different. So if you are a predator from South America, you have evolved with cane toads for tens of millions of years. And small genetic changes can actually make you very resistant to toad poisons. Or you can learn that your genes increase in frequency that say don't try to eat big, fat, warty amphibians. So you either leave them alone or else you can handle them. But that's not the case with the Australian predators. They haven't had that evolutionary history of exposure to toads, and so when the goanna or the king brown snake sees a toad hopping along at the front of the toad invasion, it thinks it's a particularly large frog and it tries to eat it and it's killed by the poisons. It dies of a heart attack almost immediately. And so we have had dramatic and absolutely tragic, heartbreaking declines in most of the apex predators in these tropical systems. The most obvious one is the goannas. The bottom right there is the yellow spotted monitor. These are big lizards and they are incredibly common before toad arrival. And the graphs show the number of goannas seen per day, caught per day. It was high until toads arrived in 2005 and then it just fell off a cliff. And the number of dead goannas we saw just suddenly went from 0 to a large number. And so in places where there are cane toads, we get something like a 95% decrease in the numbers of animals like the blue-tongued skinks, red-bellied blacksnakes on the east coast, the monitors and the northern quolls, this sort of cat-sized marsupial carnivore. So it's devastating and it completely destabilises that ecosystem because these apex predators have got a really important ecological function. So that's the bad news, and it is really, really, really bad news. But it's a bit more nuanced than that. So the first nuance is that people often say every life history stage of the cane toad is highly poisonous. And that's not true. The adults are really, really poisonous. They've got the shoulder glands with the toxin, and the female toad directs a lot of toxin into the eggs when she's about to lay them. That protects them during the egg stage when they can't protect themselves. So the eggs are really, really poisonous. But the tadpoles, as they grow older, have very little poison. It's not until they turn into a baby toad and they get their own shoulder glands and they start making their own poison that they become a real threat to native wildlife again. And these graphs just show the chemistry of all the different kinds of toxins in the eggs and then more emerging in metamorphs, but you can see the late stage tadpoles have almost none. And the other graphs are mortality rates of native frog tadpoles that eat various life history stages of toads. 100% of tadpoles die if they eat a toad egg. 0% die if they eat an older tadpole. So the older tadpoles are this stage that really has very little effect. That'll become important a bit later on. The second interesting bit is that some native species are really not affected at all by cane toad arrival, and in fact they benefit from it. We don't hear much about them, but if you're from a lineage, an evolutionary lineage that evolved in places where other kinds of toads occur, you probably can deal with the toad's poisons. And that's true for some of our harmless snakes. It's true for most of our birds. It's true for many of our rodents. So that, if we catch a native rat on the floodplain and offer it cane toads, it'll just hurl itself on them for breakfast, lunch and dinner. It's, spicy food, presumably enjoyable. They love it. And they can eat as much of it as you can give them. So for many native species, in fact, toads are not poisonous. They're just a new opportunity, a new food source. If you're an ant, well, if something kills you by giving you a heart attack, ants don't have hearts and they're not troubled by it. So ants kill millions of baby toads and are not affected by the poison. The good news about the mortality of native predators is that it's really only the big guys that get killed. The small native predators will eat toads and they cannot deal with the poison, and some of them will die. But most of them will eat a toad that's small enough that it has very little poison. And so that toad will make them really sick, but it won't kill them. And there's a process that we're all familiar with called conditioned taste aversion. If we eat something that makes us nauseous, makes us really ill, we'll remember the smell and the sight of that food or drink, and we will stay away from it. When I was a postdoc, 50 years ago, Terry and I had a friend over for a party and he brought eggnog that was off, and we were desperately ill. I have never been able to drink or eat eggnog ever since. I mean, it's an incredibly long-lasting effect. And these animals, like frogs, fish, little marsupials, they're exactly the same. They learn. So if you give them a small cane toad and it makes them sick, if that happens 2 or 3 times, they'll simply stop trying to eat any kind of a cane toad. So they're not at risk, and their populations are not affected. Okay, so that's the basics. That's the problem. Toads are killing big predators. What do we do about it? And my team has developed 2 approaches. One of them very new, one of them probably a decade old now, but it's taken a long time to bring it all the way from the idea through to implementation and adoption by wildlife agencies. And that first idea is that it may be really, really difficult to get rid of all the toads, but even if we can't get rid of toads, we might be able to make the wildlife better able to coexist with them, or to be able to buffer the impact by teaching predators not to eat cane toads. This is going to be critical. It only takes 1 adult toad to kill a predator, and there's not really all that many predators in a given hectare of tropical real estate. So you really have to either get rid of all the toads or work out a way to enable predators to survive despite the presence of toads. This is a poor goanna that we were radio tracking that ate a toad. We found dead. So we can try to make these native species more resilient. I've already said that the small species have a way to deal with toads. They learn not to eat them. But the big guys aren't saved in the same way because the toads that they try to eat are too large and have too much poison. So that if you're a small predator, like a green tree frog, they'll happily eat a small toad, but after they eat a few of them and they've got sick, they'll stop eating them. That's fine. But large predators that eat a large toad will have so much poison that it's not a learning experience, it's just the last experience of their life. They die of a heart attack. So the problem for large predators involves the invasion process. Toads come in a very large variety of sizes. So the picture there at the top, we've got a baby cane toad sitting on top of a mum. So there's a huge size range. And the toad invasion is moving incredibly quickly through tropical Australia. The toads are moving something like 1 kilometre a day during the wet season. It's an amazing phenomenon. No frog in the world moves like cane toads in Australia. They've changed their size and shape and leg length and behaviour and physiology to become dispersal machines. So the only toads at the invasion front are the big guys, the really mobile guys. And so if you're a predator like this goanna, the first toad that you're going to meet is going to be a large toad. And if you eat toads and you try to eat it, you're dead. What you want is to create a situation where that predator meets a small toad before it meets a big toad. Because that way it gets a chance to learn, and when the big toad turns up, the predator says, oh no, look, I know that you're not acceptable tucker. So we set out to test that idea. I thought it was at least worth a try, even though it sounded unlikely. We started out with northern quolls. They were being raised in a zoo outside Darwin because quolls were becoming very, very scarce in the landscape. So the zoo was breeding them up and releasing them. And we convinced the zoo to let us train half of their baby quolls on very small toads. So half of the quolls got to eat a small toad, or 2 or 3, got sick and learnt not to eat them. The other half of the quolls, their brothers and sisters, weren't trained. And then we released them all with radio transmitters, and the average guy that wasn't trained, especially the males, were dead within a day or 2. After all these months of being raised by the zoo, as soon as they met a toad, they tried to eat it and they died. Females were smarter, but if you train them, both sexes survived very well. They'd see a toad and they'd leave it alone. So it worked. The next step, so a woman called Stephanie O'Donnell did her honours on that. Sam Price-Rees did a PhD on blue tongues. Same story, except this time Sam caught blue tongues in the wild in the East Kimberley, gave them a toad meal and then let them go again. The controls didn't get a toad meal before they were released, and sure enough, when the toad invasion came through, the untrained blue tongues all died. We couldn't find a single one, but the trained guys survived quite well. But the really ambitious one was by a woman called Georgia Ward-Fear, who's doing a PhD with me, also in the Kimberley, on the Yulmbulgari floodplain. And we managed to work with the indigenous rangers, the Balangara rangers, who know that landscape incredibly well. And Georgia and her team managed to catch and radio track 150 of these giant lizards, which is not bad. And half of them were offered a small live toad. So they got a learning experience, just one toad. And the other half were not. And when the toad invasion came through, the red line is the mortality. All of the untrained guys were dead within a few months, and quite a few of the trained guys survived. So it worked beautifully at a landscape scale with free-ranging goannas that had been exposed while they were still running around being real goannas rather than sitting in a cage. And I'll digress slightly because I think it's a really interesting story. There turned out to be an extraordinary value in working with the indigenous people, with the Balangara Rangers. The way we did it was we had quad bikes on this very remote floodplain, and we had one Western scientist who was a reptile expert in every case, and one Balangara ranger. And they went together for safety reasons, obviously, and we recorded who saw the goanna that we ended up catching and bringing into the study. And it turned out that the rangers were much better than we were at seeing the goannas. They were seeing goannas that were in the shade and so much less obvious, and were motionless and so much less obvious. So extraordinary ability of these indigenous rangers to spot these animals. And when we did some behavioural tests, we discovered that the goannas that the rangers found were shy animals. They were much less bold. And they were the guys that learned from meeting a small toad. The big bold blokes, the ones that the Western scientists were finding, were big and bold and stupid. But they were ready to have a go at anything. And yes, they'd have a toad and they'd get sick and they might not eat toads for another few weeks, but when a toad hopped up a month later, they'd grab it and they'd die. But the shy toads that the indigenous rangers were finding were much more likely to remember. They remembered for much, much longer, and they were the ones that survived. So if you look at the bottom, the right-hand ones here, these are the trained guys. Most of the goannas that the rangers found were ultimately killed by being eaten by pythons, whereas most of the toads that goannas that we found were mostly killed by eating a toad. So a huge difference, and that whole project would have failed if we had just gone out there in the conventional way and not involved the Balangara Rangers. So the Balangara Rangers are authors on all of our papers, and they've just been fantastic. Anyway, that's a side thing. The whole point of the idea of releasing small toads in front of the invasion is that a single generation of education may be enough. The toads that first arrive don't breed for a year or two. So there's no small toads in the landscape. But within a couple of years, toads start breeding and the edge of the billabong is covered with small toads. So if any predator, olive python, yellow-spotted monarch, lives long enough that their babies grow up in a world where there's lots of baby toads around, then they can learn for themselves. So you don't have to repeat the education every generation. It should be permanent. You just need to buffer that 1 year of impact, and then it should be okay. So we had something that we thought worked, but the difficulty was that what I was saying was that in order to fight the problem of cane toads, you've got to release more cane toads, and you've got to release them in advance of the invasion front. And so the newspapers in Darwin and many other places thought that this was probably the most stupid idea that any human being had ever had. And you know, these bloody southern academics, what the hell do they know? This guy wants to release more toads in advance of the front. So we put an enormous amount of effort into a PR campaign for the teacher toad concept. You know, if I ever got an award, I'd talk about teacher toads. Here's Georgia and Uncle Bertie, one of our ranger colleagues. One of the radio stations flew them down to Melbourne to do a radio scare on it. This is from a documentary movie they made about the toad work. It's a book that I wrote. We really tried to get the word out that teacher toads work. They're not such a silly idea after all. And somehow or other, that just changed the whole discussion. So that by the time we were seriously talking with the management authorities, the government agencies, nobody was complaining. And so we were able to work with the Indigenous people and the government agency in Western Australia to do this at a landscape scale, to actually give it a try. So in the Kimberley, working very closely with these communities, so we're breeding up toads in the Indigenous communities, very remote areas. We've got a very robust experimental design. We're looking at the number of goannas, and we measure that with camera traps. You open a sardine can and you put a remotely triggered camera trap, and when a goanna comes along, it takes a photo. So we know how many goannas there are in the area, and we do that before the toads arrive. And then in half of those areas, we do the teacher toads, and the other half we don't. So we have a really robust idea as to what's going to happen. And this is the number that blows my mind. We released almost 250,000 cane toads in advance of the front over the last few years, and nobody complained. And it worked. The top graph, the dotted lines are the control sites where we didn't release teacher toads, and the numbers of goannas went to zero very quickly when the toad invasion arrived. And in the solid lines, which are the teacher toad sites, the goanna numbers rebounded. So goannas are still abundant in those areas despite the arrival of cane toads, whereas everywhere else across tropical Australia, they've disappeared. So it actually worked, which, you know, you don't get very many chances to bring an idea all the way through from you know, that sort of wild, crazy, hairy, God, that seems unlikely, through to actually getting it implemented. It's now the main strategy that the Western Australian Department is using in their toad control work in the Kimberley. So that's been great. At the same time, pretty much, we did the crocodiles. The freshwater crocodiles were starting to show this same wave of mortality as the goannas do in these beautiful billabongs. We used a different method. Rather than live toads, we strung up bits of toad over the water so that the crocs could come along at night and grab them. And just to make it really convincing, because crocodiles are big animals, we injected a chemical, lithium chloride, that makes you just desperately nauseous. It's not dangerous, but it really is special. And so these crocs had stomach aches like you wouldn't believe. And sure enough, in the control areas where we didn't do it, the dead crocodiles kept floating to the surface. And in the areas we did it, not a single dead crocodile. And that lasted for years. So it's worked. It's worked beautifully in both cases. So we can buffer the impact, but that's not the same as getting rid of cane toads. Australia would be a much nicer place if we didn't have cane toads. Sorry guys, it's the truth. So what are we going to do about it? Well, the traditional thing to do is to go out there and pick up cane toads at night in your backyard. And if you're humane, you pop them in the fridge, and then a few hours later you put them in the freezer and they die a humane death. This is young Marla in Darwin, daughter of one of my postdocs, Chris, very proudly holding a cane toad she's picked up. And this is kind of a mainstream activity of tropical culture, particularly in the territory in North Queensland. People go out there and they get huge numbers of toads. There's groups that form and do their big toad busting events. There's traps for toads. There's all sorts of approaches. They get huge numbers of toads because toads sit out in the open. They're advertising their poison. They're not hiding. They're not running away the way that native frogs do. They're just sitting there, and you can walk over there and pick them up most of the time. And you can get a heck of a lot of toads, but the problem is, well, a number of problems. There's a lot of toads there, and they're moving around a lot, but the big issue, the reason that this simply doesn't work for toad control, is the reproductive rate of cane toads. A single female toad, when she has her eggs, can have 20,000 eggs, maybe 30,000 eggs. And that number's a killer. Okay, you could go out and get 95% of all the adult cane toads in the local area. And then tomorrow night, it rains and the few that are left breed, and each of the girls has 20,000 kids, and you've got more toads than you started with. So you simply cannot control cane toads unless you can stop them breeding. And that was the key insight. I knew that the only method that I could sensibly look at to try to come up with a way to control toads was something that could stop them from breeding, which at first sight seemed impossible. But evolutionary theory sort of comes in and helps a bit here. One of the characteristics of most of the invasive species, including cane toads, is that they don't really like sort of the beautiful, natural, pristine habitats. They like disturbed areas. Toads like places that humans have trashed. If you go out at night and you find a cane toad, the next thing you'll find is probably another cane toad. There's not going to be very many native frogs or anything else in that area. And so cane toads in Australia reach very much higher densities than they do in their native range, as is commonly true for invaders. And their biggest enemy is other cane toads. So you've got a situation where every generation there's really intense selection to try to do the other toad down. Any mechanism that would enable you to suppress other cane toads would be terrific. It would make life wonderful for you and for your kids. And so maybe we can look for mechanisms that have evolved that enable invaders to suppress the numbers of their own species because of that intense competition. And a postdoc called Michael Crosland was the guy that really made the breakthrough. Michael is an expert on tadpoles, and this is the results of one of the experiments he ran. He discovered that cane toad tadpoles love to eat newly laid eggs of other toads. And so in this experiment, he's offered them the eggs of lots and lots of different species of native frogs, and the cane toad tadpoles really don't eat them. They're not very interested. They'll eat one or two, but they eat 100% of the eggs of cane toads. They love to eat the eggs of their own species. The reason that they're so selective is that the poison that the female toad has put into the eggs to protect them begins to ooze out as the eggs break down, just before they hatch. The egg stage is only a couple of days long. And as that poison oozes out, it triggers older cane toad tadpoles who detect that chemical to go racing across towards it and to start eating frantically. So it's a really species-specific response. That leads to incredibly high rates of cannibalism in the field. This was an experiment. You see mortality rates are 100% if there's any tadpoles in the pond. This is one where we put some eggs inside a mesh container, and these are all cane toad tadpoles that are frantically fighting their way trying to get in. So that you can literally put toad eggs or the poison from an adult toad in a container and stand back, and you'll see a stream of cane toad tadpoles starting to come towards it. Incredibly selective trapping. The tadpoles of native frogs go the other way. They're not attracted to a poison, but the cane toad tadpoles are. As soon as we knew that, we knew we had a way to trap cane toad tadpoles. Oh, okay. Second step is that Michael also ran some experiments showing that cane toad tadpoles also produce chemicals that will diffuse, say, through that mesh barrier, so that the eggs here at the bottom, because they've had those chemicals during their development, will very likely die within a few days of hatching. They won't develop teeth or tails, or, you know, they really are developmentally utterly disrupted, whereas the eggs over here will be absolutely fine. So there are chemicals produced by cane toad tadpoles that disrupt the development of cane toad eggs as well. And they also compete with, older tadpoles compete with younger tadpoles very effectively. So the result of all that is that a clutch of toad eggs that's laid into a water body that already contains cane toad tadpoles is very unlikely to survive. They're all going to get gobbled up or chemically suppressed. And the question is, how can we exploit that competition? And the simple way to do it is to take a very simple design of a funnel trap, just like people use minnow traps, they call them in the US. We put toad poison in as the bait, and it catches vast numbers of toad tadpoles and nothing else. And we've done lots of experiments on this, and basically you catch all of the tadpoles within even quite a large pond very quickly, within a day or two. We managed to interest a non-profit environmental company, Watergum, in this. We handed the patent across to them, and they have rolled this technology out right across the range of the cane toad in Australia. You can buy Watergum traps and baits, so you don't have to handle the poison yourself. And that has been incredibly successful. Millions, and I mean millions, of cane toad tadpoles are being removed from natural ponds across Australia every year using the Watergum traps with our trapping method. So that's great. The problem is that it's a lot of work. You've got to put the traps out, you've got to check them, you've got to get rid of the dead tadpoles, you've got to rebait them frequently and all that kind of stuff. So, what else could we do? Well, let's recap. What do we know about the system here? We know that toad tadpoles are not poisonous, unlike eggs or adults. So they're not a threat to native fauna. We know that they're incredibly good at controlling toad reproduction because they eat toad eggs and they produce those chemicals and they compete with smaller taddies. So they're the perfect control agent. A toad tadpole would be the best way to control cane toads. The trouble is that very helpful tadpole will reach the end of its tadpole life, it'll turn into a baby toad, and it will then start to grow its own poison, and it becomes part of the problem. What could we do about that? That's what the gentleman over there come in. The cane toad life cycle is very simple. We've got our adult toad, lays eggs. In a couple of days they hatch. The tadpoles are very small, and we've got some here to show you. They live for a couple of weeks and then they transform into baby toads. But suppose we stop them from metamorphosing. Suppose we created a tadpole that was unable to make the transition from being a tadpole to being a small toad. We call them Peter Pan tadpoles, after the fairy tale character that never grew up. And the idea is that if we could create a tadpole like that, we could put them in a water body and they would sit there and they would eliminate recruitment by cane toads as long as those tadpoles were alive. They would eventually die of old age. They're not a threat to the fauna. It could be a really selective way of stopping toads from breeding. Translating that into practice was something so far beyond my meagre abilities that it's almost laughable. So what we do is we find people like Michael and Alex that know about CRISPR-Cas9, and I'm not even going to try to explain it, except to say it's sort of a molecular scissors. It's a chemical you can inject, say, into a toad egg that will target a very specific part of the toad genome and will cut the DNA at the exact spot that you tell it to. And we hear a lot about genetically modified organisms, GMOs. People use CRISPR to put new genes in, but we're not doing that. We're simply going in and wrecking an existing pathway. We're simply creating a tadpole that can't metamorphose. And we can do that very easily because we know that the way that a tadpole transforms into a baby toad relies on a particular hormone called thyroxine. We all have thyroxine, we use it for different things. But what happens in a tadpole, any frog, is that it begins to produce thyroxine at the end of its tadpole life. That thyroxine initiates a series of changes, which means that it resorbs its tail, it starts to grow legs and lungs and all that kind of stuff. It turns into a toad. So that if we knock out the genetic pathway that enables it to make that hormone, it can't transform and it will stay as a tadpole for all of its life. There's a lot of concern about genetically modified organisms, so I really want to hammer this one. This is not a GMO. This is a buggered up tadpole. Every clutch of cane toad eggs that's ever existed in the history of the planet has probably had a few sad little tadpoles at the end that didn't manage to transform into the toad stage because they had a mutation in the thyroxine pathway or something similar. We're simply making that happen at an industrial scale. It's something that's already out there. It's an entirely natural process. And for that reason, it's not prohibited and regulated in the same way that genetically modified organisms are. Because there's no risk. These guys are going to be, you know, they're sterile, non-reproducing little tadpoles. They're going to finally die of old age. They're not going to spread. They're not going to go anywhere. And so we already have permits from the state regulatory authorities in New South Wales, Northern Territory and Western Australia to release these guys. Whereas if they were transgenic and had, you know, some novel combination of genes from somewhere else added in, this would be a nightmare and we would probably never be able to release them. So this is a really simple gene knockout. This is Michael staring down a microscope, doing it again. We induce breeding in a pair of toads by hitting them with the right hormones. It's very straightforward. Michael then has his magic chemicals. He then very, very carefully injects eggs. We then have to raise them. We have a major, large-scale tadpole raising facility. So we have this field station up near Darwin, and thank you very much, David, for your support over the years in making this possible. And it means that we can raise thousands upon thousands of our tadpoles. This is Alex Funk, the PhD student that's done lots of the work to show that Peter Pan tadpoles are not risky for the native fauna. Jamie's a volunteer. This is Quill, who's the daughter of Georgia, who's the postdoc you've already met when she was training goannas not to eat toads. So the first target that Michael attempted was skin colour. I suspected this would take a couple of years. It took him about a week, I think. The idea is that we needed a trait to see if the CRISPR method worked. And so we needed something that you could just look at an animal and you'd know whether or not you'd knocked out of the gene pathway. And so colour was a good one. And so tyrosinase, you knock that out, the animal can't deposit the pigments in its skin, and so it's an albino. And sure enough, the eggs that Michael injected turned into albino tadpoles, turned into albino toads. When they bred, they produced albino eggs. So we have changed the germline. We have created a heritable line, a strain of cane toads that are entirely albino. That was exciting. We even got an albino toad on the front page of a journal just a couple of weeks ago with the first paper that Michael's written out of this work. The next target was Peter Pan's, and again, I thought this was going to be tough, but Michael got it to work almost immediately. So this picture shows a normal tadpole in the process of, you know, metamorphosing. He's getting quite big back legs and so forth. He'll be a baby toad before long. This is a Peter Pan. He's just kept growing and he isn't metamorphosing. He's just living there in the larval stage. And we have normal wild type tadpoles and Peter Pans here. And it still takes my breath away to look at those two tanks and think that you can do this with injecting some chemicals into a toad egg. The Peter Pans, we were worried they wouldn't be cannibals, unlike the wild type, but in fact they're voracious cannibals, and because they're bigger, they eat a hell of a lot more. So they're incredibly good at eradicating a clutch of toad eggs. Are they safe? We've done a lot of work, about a decade's worth of work on the impact of normal toad tadpoles. So we were pretty confident that they wouldn't be a problem, but Alex Funk put out trays with Peter Pan tadpoles and other tadpoles and fish in among all the water birds at our study site. And the birds don't really like to eat toad tadpoles. They taste bad, they spit them out mostly, but they certainly don't get sick. There's not enough poison to make any of them bing. Turtles, fish, aquatic insects, we've run 1 million trials with, I'm exaggerating, okay, but we've run a lot of trials. Alex has run a lot of trials, and there's no problem. Mostly the predators don't really like to eat them, but if they do eat them, they're fine. So it appears to be incredibly effective and incredibly safe, environmentally incredibly targeted. Turning this into a real control strategy, though, means that we need a lot of them, and even though Michael's a hard-working bloke, we can't possibly get people like Michael and Alex to be injecting enough eggs to give us the Peter Pan tadpoles we need. But there's a simple way around that. Remember, these CRISPR-injected tadpoles can't metamorphose because they don't have the hormone. So we just put the hormone in the water in the lab. And they don't care, the tadpole doesn't care where the hormone comes from, so it will metamorphose. It'll turn into a toad, and we've got to supplement the hormone as it gets bigger, because it will need it as it grows. But in the lab, it means that we can potentially grow these guys with the Peter Pan characteristic up to adult size and breed from them. And as soon as we can do that, we've got our 20,000 Peter Pans, bang. And we don't need anything like that number for an average sized waterhole. So we can scale this up. We still have to overcome the technical issues. We're still playing with that, but it's looking very encouraging. I think we'll certainly get there. When we start thinking about letting them go, it'd be nice if they were distinctive. So that, for example, the community group that was going out there to bash toads on the head could say, oh no, look, that's a good toad, we'll leave that one, but we'll take the other guys that aren't modified by the CRISPR method. And so we've been playing with what colours to use, and the results are here in front of you. We want a colour that's distinctive, easily recognised. I think it'll also make people much more comfortable about having modified toads if they can say, oh look, that's a wild type and that's a modified one. And so we still haven't, nailed that down. We know albino's probably not going to be it. They don't do quite as well as the wild type. These leucistic guys that have black eyes, yeah, that's looking a bit more encouraging. Alex and Michael have recently produced black ones, but there's a whole range of things you can do. So we will come up with a funny looking toad that is distinctive, and I'm sure that that's doable. So guys, we're into sort of science fiction here. I mean, this is an extraordinary situation, I reckon. We are at the threshold of being able to produce large numbers of toads. Alex is knocking out toxin production pathways. So he's going to produce toads that are far less poisonous. So that if you eat one of those, even if it's a big one, you eat one of those, you'll get sick, you'll learn not to eat toads, but it won't kill you. That will be a far more effective teacher toad. At the moment, the problem with our teacher toad program in the Kimberley is that if any of our toads we release don't get eaten, they will grow large enough to eventually become fatally poisonous. But if we use Alex's little babies that never get toxic enough to be deadly, that'll be a much safer way to go. So they'll be funny looking, to be distinctive. They'll be toxin reduced. We'll have the Peter Pans in the water bodies, hopefully living for, at the moment they live for a few months, so that's enough to be useful. It'd be lovely if they lived a bit longer. But it's a whole different approach to pest control that anyone's ever tried. And this really is looking like it could be the first actual control for toads in Australia. But toads are the classic example of scientists screwing up. The Queenslanders, and I'm a Queenslander, the Queenslanders that brought cane toads to Australia in 1935 thought they were doing something responsible and sensible, and this was going to be great, solve the problem, wouldn't be any collateral damage, and it was a catastrophe. So we're moving very slowly and very carefully. We're checking all the impacts of everything that we produce and do. That means it's a bit slower, but it means that we can be very confident of what we're doing. And what we're going to do for the field tests is we're moving down to the desert fringe of the toad invasion, where they only live in these artificial water bodies, the turkey nest dams. There's a lot of toads, but they can't go out into the surrounding landscape because it's just too dry. They die within a day. So we can put Peter Pans in these water bodies and see if they do indeed block toad recruitment and if they have any other effects on anything else we can measure. And we're hoping to do that next year. It's getting very close. This is Georgia and Quill and I processing toads late at night down in the Daly Waters. We've been going there now for a few years. We're getting a lot of data before we do anything, so that again, we get a very robust picture of how we're changing the population structures and the biology of toads. as we go about doing it. The genetic implications will be particularly interesting. So that's the end of the story. Toads have devastating ecological effects in Australia, but it's primarily by poisoning these big predators, not by any other mechanism. Understanding those impacts led us to think of conditioned taste aversion as a way to buffer the impacts, and we've actually moved that now all the way through to being a rolled out, full-scale, large spatial area control by the government authorities. And understanding toad biology and the rapid evolution of cannibalism. I haven't told you, but the toad tadpoles aren't cannibalistic in South America. They've evolved it in Australia because the densities are so great that the competition's more intense. So we're taking a really newly evolved trait, which is sort of cute from an evolutionary perspective, but it's enabling us to develop powerful new methods. And with the expertise that these guys are bringing, we really look to have a different kind of a way to try to control vertebrate pests. And I'm going to finish with this slide, and an admission that everything that I've talked about sounding like it all came from my work is in fact the work of a whole bunch of people. This was from a workshop we had up at the Macquarie Uni Research Centre up near Darwin. I've mentioned Georgia a few times, a partner Miles, who also works with the project, works for the government department, and Quill, who supervises everything. Chris Jolly's a postdoc doing lots of evolutionary stuff with the toads. Greg Brown's a postdoc who's been managing the research station for 25 years. Michael's sitting over there. Alex Funk's the PhD student that's trying to check that we're not doing anything stupid. Anthony Waddell's a brilliant synthetic biologist, works with frogs, and who's been really important in the framing of the project. Marcek Misalko, one of my colleagues here at Macquarie, is the great guru of CRISPR. Alex is also sitting over there. He's doing the toxin reduction stuff. And this is Melanie Elphick. She and I celebrated our 30th year together as she's my research assistant, and she is the heart and soul of the entire research operation, the way we've been able to run things. So I am deeply grateful to all of these people for making me look smart. And thanks to the photographers whose pictures I've used, and I think that's the end of the story. Thank you.
And that was an amazing story. Fascinating journey that you've undertaken over the last couple of decades. So we do have time for some questions. Anybody? Thank you. Yes, we'll just go on the back.
Thank you for your great talk. When you're training, you have a creature that they eat, but they don't.
I'm having trouble hearing, I'm sorry.
Wait, can you? No.
Yeah, that's better. Yep.
We might just swap microphones.
Always a strong possibility it's my hearing aids, but I think it is the microphone this time. Yep. Yep.
I'm just curious about the training. You're training individuals to eat the small potato toads, and they won't die, but they have to then be averse to the big creatures. Now, do they have to taste the big creature? Is there a common factor that they recognise between small?
Yeah, look, it'd be lovely to know more about the learning process, but it seems it's almost entirely, I think, based on the way something moves and how it smells. So what we saw, particularly with the little marsupial predators, we ran the trial, little toad goes in, the antechinus races across, grabs it, eats it, gets sick, eventually, 10 minutes later, it shakes its head, it recovers, it's alive again, throw another one in, it runs across and it sniffs it before it bites. And I think the predators just take a somewhat smarter approach, and they really check that it isn't the same as that thing that they ate last time. So a big toad presumably smells a lot like a little toad, and it probably moves in a similar way. I think that's all that's happening, but we haven't actually specifically looked at it.
Just one more, if you don't mind indulging me. Cane toads for humans. Now, I think I know not to
Yeah, it's very rare for them actually to sort of, have a spray of toxin, but what happens if they get really upset, and that usually only happens when something is trying to eat them. I mean, just handling them, they very rarely exude toxin. It's a bit weird, some populations do it more. But yeah, so this white milky fluid starts to appear, and if you get any of that on mucous membranes, that's when the problem starts. So that's why the vet tries to irrigate the dog's mouth with the water and all this sort of stuff. My advice is don't eat them.
I don't think this is working. I'll shout.
That's the spirit.
So, you've already talked about the fact that the toads have phenomenal reproductive rates and that they have adapted to Australian conditions. So how much do you have to keep ahead of their ability to adapt to what you're doing to them, given you're introducing a very strong selection pressure on the population?
Yeah, look, absolutely. It's one of the critical issues for any long-term program at controlling an invasive species is that you are imposing a selection pressure and they will evolve in ways that evade it. So we're trying to pick strategies that it's going to be very hard to get out of. So for example, you could have a mutation so that mum didn't put poison in the eggs, in which case the Peter Pan tadpoles wouldn't detect them. But everything else in the pond would gobble them up because they're not protected by poison anymore. And so you're trying to find really quite central aspects of the biology of the beast that aren't easy to change for them to get around. But look, it's absolutely true. You're up against a flexible antagonist, a formidable adversary, and you've got to keep your eye on the game because these things are evolving in front of us. Toads are evolving to deal with cold climates, with aridity. A whole range of traits are changing, and we have to keep watching it.
I just got a question here. How far south are they at the moment?
There's none in the Antarctic that we've heard of. I read about a guy that in Auckland who found one in his hiking boot when he got home. But they do stow away everywhere. So our estimates are something like well over 100 toads arrive in Sydney every year. And they usually die a lonely death, but occasionally it's a group of them. There's currently a population at Lake Macquarie that's spread. Don't seem to have spread enormously, but we don't really know. You don't normally notice them until they've been around for a couple of years. We had a big population near the airport in Sydney a few years ago, and it took us 5 years to get rid of them. We had to radio track toads, find out where they were breeding, and then we could block off the breeding ponds and so forth. This was before we had the Peter Pan technology, and we haven't seen toads there for a long time. I think there's so many black rats living in the sewers that they were cleaning up the toads for us, so we had some help. But yeah, the main front is sort of near Grafton, moving quite slowly depending on weather conditions. So it'll probably be a long time before the main front reaches Sydney, but we will continue to get these incursions of the stowaway toads, and that's true throughout their range. They're very tough. There's stories about a few toads in an old Model T Ford that was shipped from Florida across to London, and by slow boat. And when they unpacked the car in London, the toads hopped out looking quite fresh and happy. So yeah, they're tough.
My question was about the colour change idea. So if you modified them to have a different colour, isn't it possible that they have like a disadvantage in reproduction because they're so opposite to other toads? Like, I know that happens in some animals.
Yeah, look, we've looked quite a lot at reproductive behaviour in toads, and the one thing that you could not call a male toad is choosy. They frequently amplex our hands or feet, and we just actually ran a whole bunch of trials with styrofoam models of different sizes and shapes and colours, and the male toads grab absolutely anything. So I don't think that'll be an issue. I think there'll be more of an issue about, like David was saying, with viability and fitness, and they need to be able to function in the wild in the face of predation and things like that. Maybe if we change skin colour, we may change something about glands in the skin that are affecting microbial peptides. We need to look at all of that stuff to create the perfect toad that will carry the construct for us.
How are they disposed? Like what happens to the they're poisonous, right?
So, yeah, what was?
The toads
Yeah,
by people.
yep.
How do they dispose of them? What happens to the toxins in those frogs
Yeah, so
or toads?
one of the very sad and unfortunate things, and this is embarrassing because Alex knows 1,000 times more about toad poison than I do, but it's incredibly stable. So we ran some experiments where we picked up some roadkill toads at the end of the dry season near Darwin that had been on the road flat for probably 7 or 8 months and run over by road trains, every half an hour. And these things are like pancakes. And you put that in water, it swells up and the toxin is still effective. So the advice to people is to bury them somewhere your dog's not going to get to them because the toxin is incredibly stable. It will be broken down by bacterial degradation in the fullness of time, but just simply lying out there in the sun, unlike most toxins, it retains.
What about the soil?
The what? The soil, yeah. Look, people say that's really good fertiliser for your roses. I don't know. But that is certainly the recommendation, and I would think that, I don't know, microbial action in the soil would break them down.
I assume it'd be higher, but just out here.
Yeah.
So there's really more moisture trapped in the soil as well.
Yeah.
So it'll help microbial process.
Yeah.
Killing them too, you wouldn't just bang them on the head with a spade.
Perhaps because the toxin would spread. Is that right?
I wouldn't bang them on the head because I actually respect cane toads. It's not their fault they're here.
Yeah, of course.
These are beautiful creatures in my eyes, and it's our fault we brought them here. They're being killed. They're killing native predators that try to kill them. So really, let's treat toads with humanity. And all of our research, we actually measured brain activity in toads that went into the fridge for a few hours and in the freezer. No activity, they feel no pain. That's the good way to do it. If you had to do it by hitting them on the head, hit them really hard, but I would be wearing eye protection as well, because it certainly is the case that if you hit the parotid glands, you're in trouble.
And how long would you leave them in the freezer? How does that work? Sure.
Until they're well and truly dead. There are many stories of people deciding they needed space for the ice cream and bringing the toads out, and they start sort of moving like a horror movie. So yeah, I mean, a few hours in the fridge and then at least a few days in the freezer, so they're well and truly toadsicles. And at that stage, I think they can
So
be
basically
disposed of.
in the fridge first, just to numb them, and then freezer. Right.
Basically,
Okay.
these cold-blooded animals are used to cooling down every day. So it's not, if we were cooled down like that, our bodies would be fighting it, and it would be incredibly stressful and painful. But for a lizard or a snake or a toad, they cool down every night. They're used to it, and by the time, that they're so cold pretty quickly that they can't feel a thing. And then you can put them into the freezer, and it's all over.
Okay, thanks.
One more question up the back.
Sorry, my hearing's terrible.
Do cane toads have any distinct sounds?
Do they have any?
Distinct sounds.
I've
Do
completely
cane
failed
toads
to
have
get-
any distinct sounds?
Sound? Yeah, the males call. It's like often compared to a motorbike or a machine gun or the dial tone of an old phone. Yeah, it's a very distinctive call, and some of the people that have designed traps have shown that if you experimentally manipulate the call to have a lower frequency, it's sexier, and the girls will come in from a longer distance to it and so forth. But yeah, that's about the only time. The boys also make a very high-pitched squeaking call called the release call when you grab them, because they're constantly, because of this sort of preparedness to have a go at anything that looks like it might be a toad, they're constantly grabbing each other. And they have a specific release call that tells the male, hey, no, I'm not one of those, leave me alone. And so if you get a lot of toads together, you'll hear all these release calls, squeaky, 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 all over the pond, because they're constantly making mistakes. The girls tend to stay away because it's dangerous for them. The boys will often drown girls that come down to the water just to get hydrated. It's a tough world out there around these turkey nest dams.
We probably have time for one more question. Okay, 2 more questions.
Yeah, because we want people to come down. Yeah.
Hi, Rick. Thanks so much for that. It was incredibly interesting.
Can you tell us what happened in the
nobody's worried about toads in Brazil because they're native species. Nobody's worried about toads in Hawaii because there's no native predators that would eat toads. And in fact, most of the toads cane toads have been introduced to about 40 countries around the world, but most of those places have got native toads. And so the predators have had millions of years to work out ways to deal with them. They may still cause problems, but not big problems. It's really Australia, New Guinea. There's some data from Japan where some of the native snakes have been knocked over. There are other kinds of invasive toads as well. There's a small species currently in Madagascar that's causing lots of concern by poisoning predators. But broadly, as with most invasive species, there's only money to study them in places where they're a problem. So we know almost nothing about the biology of the species in its native range. Almost all of the research has been in Australia. The ARC was kind to me and I managed to take my group to Hawaii for a few months, and a couple of postdocs went to French Guiana where the toads came from, and we've got data from there as well. But that's unusual. For most invasive species, almost all the information is in the invaded range.
And last question. Yes.
Thank you. So we know that the eggs are cannibals. When they consume the eggs, they don't become toxic over time, especially the Peter Pan ones.
Yeah, we actually thought that one of the sort of, motivation is the wrong word, but one of the advantages of being a cannibal might be to get poison so that you can make yourself more poisonous. It seems like mum allocates a whole bunch of poison to the egg and the amount just decreases through tadpole life, and maybe eating eggs would be the way to do it. But the trials we did didn't support that. So this is a very long-winded way of saying I have no idea.
That's the next research project. So, Rick, thank you very much. It's been great to have you.